Oxygen is both a highly dangerous molecule and essential to aerobic respiration. In this video, I'll talk about the chemistry of biology, free radicals, and oxygen. Elemental oxygen has 8 electrons, 2 electrons in the innermost 1s shell, and 6 electrons in the outer shell, the 2s and 3p orbitals. That leaves 2 unfilled orbitals. Oxygen is highly electronegative, which means it has a strong tendency to attract electrons to fill those 2 outermost open spaces, unpaired electrons. Molecular oxygen shares 2 electrons, with the ones on the right in purple, and I'm showing a double bond, 2 pairs of shared electrons. It's a nonpolar molecule, but oxygen still strongly attracts electrons. We can represent oxygen showing only the outermost electrons and showing a double bond here. Oxygen picks up an electron to become oxygen with a negative charge, superoxide anion radical. That electron has to come from somewhere else, from peptides, DNA, lipids, somewhere else in the cell. Each of these is dangerous. I'll show you a few examples. 1, it can donate an electron to iron-sulfur complexes and make them non-functional. 2, it reacts easily with carbon double bonds. The stages are that the O2E- adds an H+ to become this oxygen-oxygen-hydrogen. That then easily substitutes for one of the bonds of the double bond, easily takes that electron over here, typically breaking a bond with a carbon or maybe a nitrogen. That in itself causes problems, but the reaction continues. Different reaction products can include an OH, we lose that other oxygen, or the double bond oxygen, a carbonyl, meaning we've lost the bond to this other carbon or nitrogen, now we've split that molecule. C, it can cause linkages. In this case, we had an original double bond, we broke that bond, and this carbon formed a link to this oxygen, this carbon formed a link to this other oxygen. That itself isn't a very stable molecule. In addition, these are all capable of hydrogen bonding. With an additional electron at 2 hydrogens, which the superoxide anion radical can pick up, it becomes hydrogen peroxide. Again, it needs an electron from somewhere to do that. Another electron and hydrogen, and it becomes a water and an OH uncharged. That's strongly electronegative. It's very likely to attract a new electron. And that OH uncharged is a hydroxyl radical, which can attract another electron and hydrogen to become water, finally something safe. Each of these is dangerous because it's picking up electrons from somewhere. This electronegative molecule is picking up all these electrons. So let me show you some examples. Superoxide anion radical could add a double bond oxygen to peptides, sugars, bases, lipids, or an OH, or this double oxygen linkage. And the hydroxyl radical very easily substitutes for a hydrogen on a carbon. And all of those will change structure and hydrogen bonding, leading to changes in function. Or, let's say we have an excess of oxygen, or an oxygen molecule happens to pick up an electron from a peptide, from an amino acid. Off it goes, and now we have a positively charged amino acid, which has a strong tendency to share another electron, which can cause it to form a new covalent bond, potentially with the same peptide. That O2E- superoxide radical can grab another electron, maybe from a base in DNA, and now that base has a strong tendency to create a new covalent bond with a shared electron. The hydrogen peroxide from the electron and 2 hydrogens, H+, may pick up another electron from a lipid along with an H+ to become water and an OH uncharged, another radical, which can in turn finally pick up an electron and an H+ and become water. In each of these cases, whatever lost the electron has a strong tendency to try and fill that unpaired outermost shell, forming a new covalent bond. And that new covalent bond is dangerous. So covalent bond may prevent transcription, change function of a protein, connect things that shouldn't be connected. Excess oxygen, and especially oxygen radicals, are dangerous. Here's a way you can sketch this for yourself, showing the unpaired outermost electrons. There's molecular oxygen, brings in an electron to become superoxide, an ion radical. Another electron and two hydrogens, and we form hydrogen peroxide. Another electron, the third one we might be stealing in this chain of events, and we get water and the uncharged OH hydroxyl radical. Finally, another electron and an H+, and we get to water. So it's these electrons, these stolen electrons from peptides, DNA, RNA, lipids, that cause the damage. In each case, these molecules may form new covalent bonds. They're doing so to regain that lost electron. They can form bonds with themselves, proteins, DNA, RNA, or lipids. They can also form bonds with other molecules. In any of these cases, we can change function and cause damage.